Okay, so during this unit, we'll be talking about mitosis. This year, I did decide to split up mitosis cell division into its two separate units, mitosis and meiosis. Traditionally, I've taught them together as one large one, but being the topsy turvy, crazy thing that's going on this year, I decided let's break this down and let's do these in smaller segments instead. Um, and so we're gonna go ahead and just go with mitosis and go through it. Good thing about starting off with this one is most students, most of y'all are typically have a lot more understanding of what mitosis is. If you can't quite remember, I'll go. Once you hear words like prophase, metaphase, anaphase, telophase, usually y'all are familiar with that. And of course, we're going to have to revisit that information, but we're also going to be adding to it during this time. So we might be adding some things that you haven't quite seen yet. So let's go ahead and get today started. So today, what we're going to be looking at is the cell cycle and cancer. Now, we'll go ahead and let you know that these section of notes might be a little bit longer, but a lot of it doesn't really have a ton of information on it. It's a picture with some stuff. Um, but then there are some slides that do have a good bit of information on it. So kind of bear with me. These might be just a little bit longer than what we're typically used to seeing. But yes, today we'll be looking at the cell cycle and cancer. All right, and how those occur. So first, we need to know the phases of the cell cycle. So this is not the phases that you're used to hearing about with prophase, metaphase, anaphase, telophase. Yes, that would be part of it, but whenever we talk about the cell cycle, we're actually talking about the entire life cycle of the cell. Everything from it growing all the way to the time when it finally does divide again. So the cell cycle is split up into interphase, the M phase or the M stage, which is our mitosis part. This is when prophase, metaphase, anaphase, and telophase take place. Now, you notice right here beside mitosis, I put down division of the nucleus because that's what mitosis is. Mitosis, the definition of it is division of the nucleus. And lastly, we have cytokinesis, which cyto again is the prefix for cell. Kinesis is to separate. So we're talking about the actual division, the pulling apart of the cell into two separate cells. Now, up here at the top, it says interphase is the longest phase. And that is correct. Interphase is the longest phase. Interphase is split up into into three stages, G1, S, and G2. The great thing about these G's here on this, G just stands for growth. You have primary, which would be one, the first growth phase. And then secondary would be the second one, but you have that synthesis. That's when DNA is being replicated there right there in the middle. We're actually going to be looking at interphase a lot during today's information and what's happening there. All right, and so on this next slide, I have a chart for you that really helps with understanding when the phases take place and things of that nature. So if we look at where it says G1, S, and G2, if you notice if that was to be like a pie chart, well, most of that pie chart is all interface. That means that if we look at a cell in a microscope, the chances are that most likely most of those cells will be in interphase because it's so long of a phase. And then whenever we come all the way around, we finish with G2, that's whenever we enter prophase, metaphase, anaphase, and telophase, and that is part of mitosis. And lastly, this last little one right here, that would be cytokinesis, that final division. Now, whenever you divide the cell, that's whenever you have two immature cells that now enter this process of growing again. So that's actually what's happening here is that this is a cycle. They are constantly going through this cycle, okay? I think this image does a great job in illustrating not only when they take place, but which ones are the longest. So if I ask you the longest part of interphase, you can probably look at that really obviously and tell that's G1, okay? So I do think this image helps out a lot there. It also helps out in letting and helping you realize where the thing starts at, where G1 is, and then where other things would begin at. So after G2, that's when mitosis would begin. Well, after telophase, that's when cytokinesis is. So we're going to look at these stages individually and what's happening during it. So during interphase, the G1 stage, that's whenever we primarily have the growth. The cell is going to mature. It's going to make more cytoplasm. It's going to make more organelles. It's going to get bigger so that it can actually develop and get ready for division eventually. This is also whenever the cell is going to do everything it needs to do to get ready to copy DNA. Now, I need to make sure I clarify this. This is not when DNA is being copied. This is whenever the cell is getting ready to copy DNA. Making sure that it's large enough, making sure that it's adding enough cytoplasm to the cell, or it's adding enough ribosomes to sit there and make proteins, or all the other functions that need to take place first. So after the cell grows, we enter the S stage. Now, S stands for synthesis. Whenever we synthesize something, we are making something. Kind of like photosynthesis is whenever we were making energy using light, or actually making food using light. Well, here during this synthesis stage, what we're going to do is we're going to make or replicate, copy the DNA. So if this was our original DNA strand, what we would do is we would end up copying it to where we would have two identical DNA strands. And the reason why we would want two of them is because whenever we replicate a cell and we end up with two cells, well, they both need a copy of the DNA, and that DNA needs to be identical. There's the original, and those are the two identical copies. We are going to be looking into that more seriously and its fullest process of how that replication takes place during our next day's information, during day 2's information. Lastly is G2. G2, now we're just getting ready for division. We're going to grow the cell a little bit more. Animal cells need to have centrioles, so this is whenever that growth of those are going to take place. We're also going to start seeing growth of the spindle fibers as well, because those are two things that are very important during cell division in eukaryotes. So right here, I found this picture and I really liked it. The only thing we're not going to worry about at this stage, you will get into it into college biology, is if you notice down here at the bottom in that pink section, right after prophase, it says prometa. We will not be going into prometaphase. We are just going to stick with the basics of prophase, metaphase, anaphase, telophase. All right, so I really like this image because I went through there and I wrote some things down on it because I was able to do that on this one, and it actually shows the two daughter cells being made here at the end. So of course we have just the basic information. G1, the cells are maturing. S, the DNA is being copied. G2, the cells are preparing for division. How are they doing that? Making centrioles, making spindle fibers. Once that's done, now we can enter prophase. So now we can enter mitosis. Make sure you know where these go at on the chart. You know, if I gave you this chart and let's say instead of using the actual terms like G1S and G2 and having everything labeled, what if I just used letters of the alphabet, A, B, C, D, E, F, G, something like that? And I said, and instead of that S being there, there was a C there, and I asked you, well, what happens there? You're going to need to make sure that you can look at this charts like these and be able to decipher what is it asking, where things are located at. I like the fact that also down here, whenever it's talking about the meiotic phase, it goes, the cell divides into identical cells. So during mitosis, it's all about making identical cells. And those two daughter cells, bam, they're identical. And then they're going to enter this growth phase again. So because they're going to grow and then eventually divide. Okay? So hopefully this image kind of helps bring that home a little bit. So next thing we're going to talk about is during this whole cycle, there are checkpoints located. Now these checkpoints, what they're supposed to do is they're supposed to control the cell's progress through the cell cycle, make sure it's not going through it too fast. Because just like your parents are trying to do, they don't want you to grow up too fast because they know that there are things out there that you are not quite ready to know yet. Well, that's basically what's happening here. They don't want them to try to grow up too fast before they're actually ready to conquer those things. Because if they do, they may not be prepared for it and in turn become damaged. So the checkpoints are positioned throughout the interphase, throughout the whole part of it, to ensure that the DNA is going to be accurately replicated. So it's going to check to make sure it copied it correctly. You know, I know that none of y'all would ever do this because y'all are amazing students. But, some students out there cheat. Well, whenever they cheat on an assignment, if they don't accurately do it, then honestly, they might turn in something that, what if little Johnny that they're cheating on has version A of the test and I have version D and I cheat off a little Johnny. While I may have his exact answers, I don't have the answers that go to my test. So you have to make sure it's done accurately. And then you also have to make sure that each daughter cell receives the same number of chromosomes. Now, typically, right here, I usually talk about, and we'll talk about this in our next unit on meiosis. Um, but I usually talk about Down syndrome because a lot of people know what Down syndrome is, and that Down syndrome is whenever you have an extra chromosome, and we'll get into that more specifically later on. Well, that's part of those checkpoints failing is extra chromosomes got pulled across, and basically the information wasn't done correctly. That's also, of course, there's also other causes for that. You have non-disjunction, which that's a vocabulary term we'll get in our next unit whenever we go over that. If these checkpoints do not accurately examine or stop DNA replication, a problem can occur. So if it doesn't accurately examine the stuff to make sure everything was done is identical, then that means we could end up with a genetic mutation. And we'll get into that later on in the year as well. But you could also end up right there where it talks about or stop the DNA replication. You could also end up with what's called uncontrolled mitosis or uncontrolled cell division. And whenever you have uncontrolled cell division, that causes cells to become cancerous, which leads to tumors. And that's what cancer is. And there's a lot of different types of cancers, but cancer is basically you have cells that will not stop dividing. And whenever they're dividing, they're going through that process too quick because even the cells that they're producing are immature daughter cells, and they're not ready for that. And then, of course, if cancer spreads, it typically spreads using the lymphatic system and the circulatory system. That's how it spreads throughout the body. And whenever that happens, it makes it harder to stop because that means that more cells in different areas can become cancerous. And of course, this right here is an image that I found on I think on Google, and it just shows basically cancerous cells under a microscope. So here's a quick review for today. So basically breaking this one down, what is the longest stage of the cell cycle? That would be interphase. During which stage of the cell cycle, that's what CC means, during which stage of the cell cycle does cell division occur? Well, if we're talking about cell division specifically, the division of the cell, then that's going to be cytokinesis. Remember, mitosis is the division of the nucleus. What happens during G1S and G2? During G1, that's our primary growth phase. We're going to grow the cell, make the cell bigger, mature the cell. S is synthesis. That's where we're going to replicate and copy DNA. G2 is whenever we're going to finish growing the cell a little bit. We'll also make organelles and structures needed for cell division, like centrioles and spindle fibers. What does it mean for a cell to be cancerous? Basically, it means that that the checkpoints have failed and it's undergoing uncontrolled mitosis, or the DNA is constantly being replicated and there's no controlled DNA replication. How do cells become cancerous? From checkpoints failing and the DNA having uncontrolled DNA replication. How do cancer cells spread throughout the body? That would be mitosis. I'm sorry, I said mitosis. The lymphatic system and the circulatory system. And the last one here, how does the original DNA strand compare to the replicated DNA strand? They are identical. The daughter cells are identical to the parent cell, to the original cell, we should say. They are also identical to each other, so they're identical in every way. All right? So next class, what we're going to talk about specifically is DNA replication and asexual reproduction. Don't forget, you do have worksheets on this information that's on Canvas. You also have those quizzes that I've made and links to it. Please be looking over that and practicing with that, so you'll be ready for your quiz next class. Thanks.